And the first thing we have itemized on the program schedule is talking about the importance of proper sample collection in microbiology. So, in the introduction, in the opening, I've mentioned the issue of sample collection. It is very, very important in microbiology. And why is it important? Number one, microbiology in a medical setting, if the wrong sample is taken, it will affect the diagnosis they are going to do for the individual. Now, proper sample collection ensures that contamination is avoided or prevented so that it is not the contaminant that we are counting as the organism of interest that we are looking for. Another reason for for care in sample collection, why it is important, number three, is because of reproducibility and reliability. If you don't take sample properly, that means that work will not be reproducible, and that work will not be reliable. Another reason for sample collection is cost effectiveness. If you make an error in sample collection, it can lead to repeated tests. You will be repeating tests and additional investigation, and in medical setting, it can lead to delayed treatments. And all this increase the cost of taking care of patients in hospitals. But outside of hospital, environmental samples, industrial samples, and other samples, we also need to be careful. If you don't take a proper sample, it's going to affect what you're going to get. You are going to be getting false positive results. So what you say is there is not what is actually there. So the result is positive, but it's the false. So to avoid this, that is why we do proper sample collection. Now, let's talk about the common challenges in sample collection. There are some challenges that come up when people are collecting sample. Number one is improper timing. When the timing for the sample collection is wrong, it will affect the type of sample. Towards the end, I'm going to talk about the timing of sample collection, why it is important to take samples at particular times. If you are taking water sample, you take it early in the morning or later in the evening. But later in the evening, you may not have light to be able to take it, except in some cases. And why are you going to wait until in the evening, in most cases, to take your sample? So you take your sample early in the morning so that you can bring it back to the lab and analyze it. And then you can go home in the evening and go and relax. And then the following morning again, you will do your work. Reason why you take sample early in the morning for environmental sample is because the microflora of the environment are still retained. The moment you allow changes to occur, after some changes have occurred, you will see that when sunlight comes, the ultraviolet ray of sunlight is going to kill some of the organisms. So you will not be able to take the correct sample. But in the morning, all the organisms are still in their resting state, they are still in their natural state. So you can still meet them there. Then another challenge in sample collection is contaminated equipment. If what you are using to take the sample is contaminated, you are going to get wrong sample because the contaminant in the container is going to impact, is going to also take the part of what you are going to say is you found in the sample. Then another common challenge in sample collection is poor storage conditions. Temperature fluctuations or delays in transportation samples can degrade the quality of the sample. So that's why when you go for a sample collection, you go with ice chest. There'll be ice in the container, ice block in the container, inside a cooler. And then so that the temperature will be kept from the sample point to the lab. So these are some of the things that you have to do when you're taking your sample. Then sometimes in sample collection, another challenge is inadequate volume. Some people do not take the required quantity. Let's assume you want to do a sample on BOD. You want to test for BOD, dissolved oxygen at day 1 and dissolved oxygen at day 5. If you go ahead and take, there are some cases where they went ahead. I've seen a case where they went ahead and take sample in only one bottle. And you want to take BOD, you take sample in only one bottle. You are supposed to go for BOD test, go with 2 bottles. So one of the 2 bottles will be filled to the brim. Will be filled to the brim. And then when filled to the brim, one of them, the moment you get back to the lab, you wrap it in black container, black polythene bag, and put it in a dark cupboard, in a dark place, so that biochemical reactions, no biological reaction will take place again, because in the light, biochemical reaction can still take place. You fill it to the brim so that there will not be space for oxygen, dissolved oxygen in the space, because what you want to measure is BOD, talking about oxygen. So the first one, you keep it until day 5. The second one, you get to the lab, you start the analysis already, do your titration and every other thing. Then you get your dissolved oxygen for day 1. Then by the 5th day, the one you kept in the dark, you do your dissolved oxygen also for that one. But in the case whereby you go with only one bottle, what that means is that you are going to not be able to do the proper test in that case, because what you're going to be reporting will be jargons. So another challenge of sample collection is lack of standardized procedures. So lack of standardized procedures is another challenge of sample collection. Inconsistent collection protocols across laboratories can lead to variability in results. So when you are not consistent with sample collection, it's going to affect what you are doing. As microbiologists, I am sure you do not want to, or as a researcher, you do not want to contribute nonsense to the scientific world. You want to contribute something tangible, something that will be of essence to the whole scientific world. And that is why you do proper sample collection, do your proper test, and report properly. But if at the point of sample collection, your sample collection is wrong, what do you want to report eventually? You will also report something wrong. So that is why it is important to take proper samples. Now, let me tell you some best practices for proper sample collection. Number one best practice for proper sample collection is you use sterile equipment when you are going for sample collection. Use sterile equipment. Everything you want to use. Always use sterilized containers, swabs, or other tools to avoid contamination. If what you are using for sample collection is not sterile, there will be contamination and you'll be reporting the wrong thing. Another best practice for proper sample collection is follow guidelines. In every area of microbiology sample collection, there are guidelines. So find out the guidelines concerning the type of sample you want to take and adhere to it, so that you will not be going back and forth, and so that your results can be reproducible. So adhere to standardized protocols for collecting, handling, and transporting different types of samples. Don't just think that we're just doing all these things for anyhow. These things are very, very important, and they're going to contribute to the general body of knowledge or scientific knowledge, both in Nigeria, in Africa, in Europe, in America, everywhere. So that is why you follow guidelines, international guidelines. Then, number 3 best practice for sample collection is that you have to label your sample appropriately and accurately. Before you leave the lab, you label your bottles. So proper labeling with proper labeling with the name or anything that pertains to the sampling site. You will have determined your sampling site, sampling point before you leave the lab. So you label properly. By the time you get there, you just look for the bottle labeled, then you pick it, and then you take your sample, or whatever kind of sample you are taking, whether it is swab or whatever. You have labeled everything before you leave the lab, then you take your sample easily. Then, another best practice in sample collection is that you have to maintain aseptic technique. Ensure your hands, equipment, and collection sites are properly sanitized to prevent contamination. When I visited a lab in the United States, I remember my host over in that lab, my advisor, he will say I will just put little ethanol in my hand to wash my hand. He will say I should take more, I should put more, I should put more. I will put it, you will see the water like this, plenty. Now I use to wash my hand before I work in the lab. He even said that some people used to drink alcohol. So you using alcohol to rinse your hand will not harm you, because people drink it. So using alcohol, ethanol to rinse your hand before working in the lab will not affect you. So when you are taking your sample, maintain aseptic technique so that nothing is going to contaminate the sample. Then, apart from these, you have to also use appropriate transport and storage media. Use transport media and store samples under recommended conditions to preserve their integrity until analysis. Then number 6 best practice for sample collection is that you have to train your personnel. Regular training of your personnel who are taking sample is important in sample collection, so that they can be able to take the appropriate sample, just like we are doing now. This is a kind of training. Then, in sample collection, ensure timeliness. Collect and deliver samples to the laboratory promptly to avoid degradation or overgrowth of certain microorganisms. The moment you take your sample, the next thing is to go to the lab immediately. Now, I've seen people that they want to collect different samples from different places. If you are going to collect samples from different places, make sure that you go for sample collection in the morning. Now you have 5 sampling points. You go to this place in the morning, go to this place by 10 o'clock, go to this place by 11 o'clock, you get to this place by 1 o'clock. All the ones you are taking after 10 a.m., those samples are not the correct samples. So if that is the case, any sample you want to take, if you take sample after 10 a.m. from the environment, that sample is not representative. In fact, take your sample between early in the morning, early in the morning and 9 a.m. So any sample you are taking from 10, 11, 12, 1, 2, that one is not representative again. The ultraviolet ray of sunlight have already killed some of the organisms, have inactivated them. Then you get there, are taking samples. So what you are taking is not representative. It's not the actual sample you're supposed to take from the environment. Now, I'm going to conclude this by saying that proper sample collection is indispensable to microbiology. So these are some of the things. Proper sample collection is a must if you are going to do a good microbiology research. So this is where I'm going to stop for now on the issue of sample collection, and I'm going to allow other people to take their own aspect of the presentation. So I'm going to call on the next presenter to get ready and take a sample. Or do we have any question you want to ask, please? Does anyone have any question? Any question, please? Okay, in the absence of any question,